Welcome to this lecture on computational biology. Today, we are going to explore two of the most fundamental and widely used concepts in biostatistics: standard deviation and standard error. If you have ever looked at a scientific paper and wondered what those little error bars on the graphs actually mean, you are in the right place. By the end of this video, you will understand exactly how to measure the spread of your biological data and the reliability of your experimental results. Let us start by imagining you are a biologist measuring the lengths of two different species of fish. In the first species, almost every fish is exactly 10 centimeters long, with very little variation from one fish to the next. In the second species, the average length is also 10 centimeters, but you find some fish that are 5 centimeters and others that are 15 centimeters. Even though both species have the exact same average length, the second species has a much wider spread of sizes. We need a mathematical way to describe this spread, or bounce, in our data, so we can compare them accurately. Now that we see why measuring spread is important, let us define what standard deviation actually is. Standard deviation is a statistical measure that calculates the average amount by which individual data points differ from the mean of the dataset. When you collect data in an experiment, you first find the center point, which is the mean. Then you look at every single measurement and ask how far away it is from that center point. A low standard deviation means your data points are tightly clustered around the mean, while a high standard deviation indicates they are spread out over a wider range of values. This brings us to why this metric is absolutely critical in computational biology. Biological data is naturally noisy and variable. Imagine you are testing a new drug and measuring blood pressure. Knowing just the average drop in blood pressure is not enough. You must know if the drug affects everyone consistently, which would give a low standard deviation, or if it causes huge, unpredictable swings in different patients, resulting in a high standard deviation. Without this metric, we could easily misinterpret our results and make dangerous assumptions about how reliable our biological measurements actually are. Now that we understand the concept, let us look at the mathematical formula for sample standard deviation. Conceptually, this formula is just finding the average squared distance from the mean, and then taking the square root to return to our original units. Let us break down the symbols. The letter S represents the sample standard deviation. The variable x sub I represents each individual data point, while x bar is the mean of those points. The Greek letter sigma tells us to add up all the squared differences. Finally, n is the total number of data points. If the spread of the data increases, the differences get larger, which causes the standard deviation to increase. Before we move on, let us see this formula in action with a concrete example. Suppose you measure the diameters of 5 cells under a microscope, getting 4, 6, 5, 8, and 7 micrometers. We want to find the standard deviation to understand how much they vary in size. First, we calculate the mean by adding the values and dividing by 5, giving us a mean of 6. Next, we subtract the mean from each diameter and square the result, which gives us a total sum of squared differences equal to 10. We divide this by n minus 1, which is 4, giving a variance of 2.5. Finally, taking the square root gives our answer. This means the cell sizes bounce around the average by about 1.58 micrometers. So we have successfully measured the spread of a single sample. This brings us to a new challenge. Imagine you want to know the average height of all the oak trees in a massive forest containing millions of trees. You cannot possibly measure every single tree, so you take a small sample of 50 trees and calculate their average height. But how confident are you that your sample average is exactly the same as the true average of the entire forest? If you went back and picked a different group of 50 trees, you would probably get a slightly different average. We need a way to measure this reliability. To solve this problem of sample reliability, we use a tool called the standard error of the mean. While standard deviation tells you about the spread of individual data points within a single sample, standard error tells you about the spread of multiple sample means around the true population mean. It quantifies the precision of your sample mean. It tells us how much we expect our sample average to bounce around if we kept taking new samples. A smaller standard error indicates that your sample mean is likely a very accurate reflection of the true population mean. Now that we know what standard error is, why is it so vital? In computational biology, we almost never have access to the entire population of a species, a cell type, or a gene expression profile. We are always working with limited samples drawn from a larger population. Standard error is vital because it allows us to draw error bars on our graphs and calculate confidence intervals. When you read a scientific paper and see a value reported as a mean plus or minus some number, that number is usually the standard error. Without it, we would have no mathematical way to prove that our experimental findings are statistically significant, rather than just random luck. This concept leads us directly to the formula for standard error, which is beautifully simple but incredibly powerful. Conceptually, it adjusts our data's natural spread by the amount of evidence we have collected. Let us look at the symbols. SE stands for the standard error. On the top of the fraction, S is the sample standard deviation that we learned how to calculate earlier. On the bottom, N is the sample size, and we take its square root. Notice what happens physically. If our data is very noisy, meaning S is large, the standard error increases. However, if we increase our sample size N by collecting more data, the denominator gets larger, which forces the standard error to decrease, giving us a more accurate estimate. Now that we have our formula, let us calculate the standard error for our previous cell example. We already found that for our 5 cells, the sample mean was 6 micrometers and the sample standard deviation was 1.58 micrometers. We want to understand how precisely this sample mean estimates the true average diameter of all cells of this type. We take our standard deviation of 1.58 and divide it by the square root of our sample size, which is 5. The square root of 5 is approximately 2.236. Dividing these gives a standard error of 0.71. This means if we repeated this experiment, our calculated average would typically fluctuate by about 0.71 micrometers. To truly cement this idea, let us look at a visual comparison of standard deviation and standard error. On the left, you see a wide blue bell curve representing the standard deviation. Notice the individual data points scattered widely across the horizontal axis. This shows the biological reality and natural variation of the sample. Now look at the right side. The narrow green bell curve represents the standard error. It is much tighter because it shows the distribution of sample means, not individual points. The standard error will always be narrower than the standard deviation because averages fluctuate much less than individual measurements. This diagram perfectly illustrates how standard error gives us a precise target for the true population mean. We have covered a lot of ground today, so let us summarize the most important ideas. First, Always remember that standard deviation describes the biological reality of your sample. It tells you how much individual data points vary from each other. Second, standard error describes the statistical confidence of your experiment. It tells you how accurately your sample's average represents the true population average. Finally, collecting more data will not necessarily change your standard deviation because natural variation always exists. However, collecting more data will always shrink your standard error, making your scientific conclusions much more reliable. You now have the tools to confidently analyze biological data.